The cell. The cell is the structural and functional unit of life. It may be also regarded as the basic unit of biological activity. The concept of cell originated from the contributions of Schleiden and Schwann, 1838. However, it was only after 1940 the complexities of cell structure were exposed. Prokaryotic and eukaryotic cells. The cells of the living kingdom may be divided into two categories. One, prokaryotes lack a well-defined nucleus and possess relatively simple structure. These include the various bacteria. 2. Eukaryotes possess a well-defined nucleus and are more complex in their structure and function. The higher organisms, animals and plants, are composed of eukaryotic cells. The plant cell differs from an animal cell by possessing a rigid cell wall, mostly composed of cellulose and chloroplasts. The latter are the sites of photosynthesis. The cell consists of well-defined subcellular organelles enveloped by a plasma membrane. By differential centrifugation of tissue homogenate, it is possible to isolate each cellular organelle in a relatively pure form. Nucleus. Nucleus is the largest cellular organelle surrounded by a double membrane nuclear envelope. The outer membrane is continuous with the membranes of endoplasmic reticulum. At certain intervals, the two nuclear membranes shave nuclear pores with a diameter of about 90 nanometers. These pores permit the free passage of the product synthesized in the nucleus into the surrounding cytoplasm. Nucleus contains DNA, the repository of genetic information. Eukaryotic DNA is associated with basic protein, histones, in the ratio of 1:1 to form nucleosomes. An assembly of nucleosomes constitutes chromatin fibers of chromosomes. Thus, a single human chromosome is composed of about 1 million nucleosomes. The number of chromosomes is a characteristic feature of the species. Humans have 46 chromosomes, compactly packed in the nucleus. The nucleus of the eukaryotic cell contains a dense body known as nucleolus. It is rich in RNA, particularly the ribosomal RNA, which enters the cytosol through nuclear pores. The ground material of the nucleus is often referred to as nucleoplasm. It is rich in enzymes such as DNA polymerases and RNA polymerases. Hutchinson-Gilford progeria syndrome, HGPS, is a rare condition of aging beginning at birth. Incidence die in 5 million births. HGPS occurs as a result of distortion of nuclear envelope due to accumulation of abnormal protein, namely lamina A. Mitochondria. The mitochondria are the centers for the cellular respiration and energy metabolism. They are regarded as the powerhouses of the cell with variable size and shape. Mitochondria are rod-like or filamentous bodies, usually with dimensions of 1.0 micrometers by 3 picometers. About 2,000 mitochondria, occupying about 1/5 of the total cell volume, are present in a typical cell. The mitochondria are composed of a double membrane system. The outer membrane is smooth and completely envelops the organelle. The inner membrane is folded to form cristae, Latin crests, which occupy larger surface area. The internal chamber of mitochondria is referred to as matrix or mitosol. The components of electron transport chain and oxidative phosphorylation, flower protein, cytochromes B, C1, C, A, and A3 and coupling factors are buried in the inner mitochondrial membrane. The matrix contains several enzymes concerned with the energy metabolism of carbohydrates, lipids and amino acids, e.g. citric acid cycle, E, oxidation. The matrix enzymes also participate in the synthesis of heme and urea. Mitochondria are the principal producers of ATP in the aerobic cells. ATP, the energy currency generated in mitochondria, is exported to all parts of the cell to provide energy for the cellular work. The mitochondrial matrix contains a circular double-stranded DNA, mTDN, RNA and ribosomes. Thus, the mitochondria are equipped with an independent protein synthesizing machinery. It is estimated that about 10% of the mitochondrial proteins are produced in the mitochondria. The structure and functions of mitochondria closely resemble prokaryotic cells. As hypothesized that mitochondria have evolved from aerobic bacteria. Further, it is believed that during evolution, the aerobic bacterium developed a symbiotic relationship with primordial anaerobic eukaryotic cells that ultimately led to the arrival of aerobic eukaryotes. Endoplasmic reticulum, the network of membrane-enclosed spaces that extends throughout the cytoplasm, constitutes endoplasmic reticulum, ER. Some of these thread-like structures extend from the nuclear pores to the plasma membrane. A large portion of the ER is studded with ribosomes to give a granular appearance, which is referred to as rough endoplasmic reticulum. Ribosomes are the factories of protein biosynthesis. During the process of cell fractionation, rough ER is disrupted to form small vesicles known as microsomes. It may be noted that microsomes as such do not occur in the cell. The smooth endoplasmic reticulum does not contain ribosomes. It is involved in the synthesis of lipids, triacylglycerols, phospholipids, sterols, and metabolism of drugs, besides supplying calcium ions for the cellular functions. Golgi Apparatus. Eukaryotic cells contain a unique cluster of membrane vesicles known as dictyosomes, which, in turn, constitute Golgi Apparatus or Golgi complex. The newly synthesized proteins are handed over to the Golgi Apparatus, which catalyze the addition of carbohydrates, lipids, or sulfate moieties to the proteins. These chemical modifications are necessary for the transport of proteins across the plasma membrane. Certain proteins and enzymes are enclosed in membrane vesicles of Golgi apparatus and secreted from the cell after the appropriate signals. The digestive enzymes of pancreas are produced in this fashion. Golgi apparatus are also involved in the membrane synthesis, particularly for the formation of intracellular organelles, e.g. peroxisomes, lysosomes.
Lysosomes. Lysosomes are spherical vesicles enveloped by a single membrane. Lysosomes are regarded as the digestive tract of the cell, since they are actively involved in digestion of cellular substances, namely proteins, lipids, carbohydrates, and nucleic acids. Lysosomal enzymes are categorized as hydrolyses. These include the enzymes with substrate in brackets, deglucosides, glycogen, cathepsins, proteins, lipases, lipids, ribonucleases, RNA. The lysosomal enzymes are responsible for maintaining the cellular compounds in a dynamic state by their degradation and recycling. The degraded products leave the lysosomes, usually by diffusion, for reutilization by the cell. Sometimes, however, certain residual products, rich in lipids and proteins, collectively known as lipofuscin, accumulate in the cell. Lipofuscin is the age pigmentor wear and tear pigment, which has been implicated in aging process. As the cell dies, the lysosomes rupture and release hydrolytic enzymes that results in post-mortem autolysis. The digestive enzymes of cellular compounds are confined to the lysosomes in the best interest of the cell. Escape of these enzymes into cytosol will destroy the functional macromolecules of the cell and result in many complications. The occurrence of several diseases, e.g. arthritis, muscle diseases, allergic disorders, has been partly attributed to the release of lysosomal enzymes. Inclusion cell, eye cell disease, is a rare condition due to the absence of certain hydrolases in lysosomes. However, these enzymes are synthesized and found in the circulation. Eye cell disease is due to a defect in protein targeting, as the enzymes cannot reach lysosomes. Peroxisomes. Peroxisomes, also known as microbodies, are single membrane cellular organelles. They are spherical or oval in shape and contain the enzymic catalase. Catalase protects the cell from the toxic effects of H2O2 by converting it to H2O and O2. Peroxisomes are also involved in the oxidation of long-chain fatty acids, CAT, and synthesis of plasmalogens and glycolipids. Plants contain glyoxysomes, a specialized type of peroxisomes, which glyoxylate pathway, are involved in the peroxisome biogenesis disorders, PBDs, area group of rare diseases involving the enzyme activities of peroxisomes. The biochemical abnormalities associated with PBDs include increased levels of very long-chain fatty acids, C24 and C26, and decreased concentrations of plasmalogens. The most severe form of PBDs is Zellweger syndrome, a condition characterized by the absence of functional peroxisomes. The victims of this disease may die within 1 year after birth.